All right, welcome everybody. My name is Jill Averill. I'm the project manager at Keystone Symposia. Thank you all for joining us for today's Career Roundtable. So a little bit about how today is going to go. We are going to have our panelists kind of discuss their career trajectory, and then I will be asking them some questions. During this time, I'd welcome everyone to submit questions that they have for the panelists in the Zoom chat box. At the half-hour mark, I will have everyone go ahead and turn on their cameras, and then I will call on you to unmute yourself to ask your question live. So at this time, I would like to go ahead and welcome our panelists. We've got Hao Wu, Felix Rando, and Andreas Linkerman. So if you all could go ahead and turn on your cameras. All right, thank you. And you can go ahead and unmute yourself. And then Andreas, would you like to go ahead and start off and kind of talk a little bit about your career trajectory?
Yeah, sure. Jill, thank you very much for having me. And it's great to be here with Felix and Hao, of course. I've never had a session like this before, but I think it's about, the way the career trajectory and how I actually ended up being a clinician scientist in the way that I am now. So I must say I turned 44 recently, and I'm a senior consultant nephrologist. So I'm working in the hospital. I'm working in nephrology. So we're dealing with kidney transplantation, which is why we, of course, know about the work of Polly for a long time. These are bedside discussions that are going on there. We have a research laboratory as well. So for myself, it's going to be like 40 to 60%, 40% clinics and 60% research, I would say, that I'm doing at the moment. But that wasn't the case during the recent years. So 5 years ago, I moved to Dresden, and I was in Kiel before. That's just another town in Germany. And during that time, I was doing a full-time clinician job. So 100% clinical routine, and everything in terms of research was on top of that. So it's not always been so super easy to perform this. Can imagine that you would then work several hours after all the other clinicians went home. You would still go to the lab and work in the weekends, these things. So then, but that got me into this nice position to apply for a Heisenberg professorship, which is founded by the German Research Foundation. And I have that now for 2.5 years, which it's a chair, it's a full professorship that covers my salary. So the clinic doesn't have to bother about my salary anymore. And it gives the clinic this nice opportunity to still have me working as a clinician without paying for me. And on top of that, get some research output that they can put on their affiliations. The lab has evolved, we are fighting for lab space like pretty much everybody is probably doing around the globe. And we have a split lab with 3 different laboratories that are not directly connected. So we're running around on campus all the time. We're now 18 people in my group. Many of them are MD students, of course, but we have 3 postdocs, we have PhD students. We just recruited a new postdoc who's going to start from the 1st of June, and we have more positions available. So if you're interested in this work, we're of course happy to include international applicants. We have English as the working language because we have people from Italy, we have people from Greece that have really given this whole thing an international character. And so I'm somewhat working on the edge between clinics and research, and that's where the questions come from the bedside. You wonder about necrosis, about inflammation, how this is connected. And this is pretty much why we are so interested in the dams, because they're triggering transplant rejections. And as we discussed earlier in some of the talks today, necrosis can drive inflammation in a heart attack, for example, or in the brain. So we're trying to understand this interface with the idea to then develop drugs and really change clinical routine. I think that's it for now.
All right, great. Thank you.
Thanks.
Felix, would you like to go next?
Yeah, sure. Well, first of all, good evening, everybody. Thank you for having me here. So my career trajectory was very straightforward. I read biochemistry in Germany. Then I went to Boston to do a postdoc with Brian Seed. And at this point, we were interested to find actually the first pattern recognition receptors. Toll-like receptors were not known. I wanted to find the LBS receptor. That was a big goal. And I didn't want to do this in a lab that was working on this. I wanted to go somewhere where people were technically able to do this, but I did not want to be the 20th postdoc on the same problem. And with Brian, that was a perfect opportunity. And after that, I came to England. I'm now in Cambridge at the MRC Laboratory of Molecular Biology. It's now almost 20 years, close to 20 years. Very happy here. So we are interested in something that I call cell autonomous immunity as a sub-branch of innate immunity. Once we had realized that unicellular organisms, of course, do not have a proper immune system. They don't have any spare cells to build an immune system. But still, they have defended themselves from the very beginning of life until today against infection very successfully. So individual cells can sense pathogens, they can fight pathogens. And we just started wondering how much of that would be in our cells. We started looking at epithelial cells, how bacteria that invade these cells are detected and fought off. And that has kept us very busy.
All right, great, thank you. And how, why don't you go ahead and tell us a little bit about your career trajectory?
Leave the best for last time. Just kidding. So how can I start? So I actually started, learning biology through medicine. I was in medical school when I was in China. In fact, my parents really wanted me to be a real doctor. So, but then the medical school that I went to had a program, one year program in the middle of the medical training to do research. So as soon as I started doing research, I decided I'm gonna, quit medical school and do research. So at that time, I, in fact, met my PhD advisor in China. So there was a international meeting about chemistry at the time. He gave a plenary talk on the crystal structure of a virus. I was just blown away by that. I've always been really interested in physical science, physics, mathematics. So that was, for me, was a awakening moment. I was like, oh, this is something, I really want to do this, because it's solving some biological questions with the skills that I think I can learn. So that's it. So I went to work for him. That's Michael Rossman. If some of you may heard of the Rossman fold, he's a really a luminary in the field. That was the point of no return. Did PhD with him on virus crystallography, and then I went to Columbia University to work with Wayne Hendrickson, who works on receptor proteins at the time. So I worked on HIV. I worked on the CD4 receptor, which is a receptor for HIV. So in that sense, I was a really hardcore crystallographer by the time I finished my postdoc. But when I tried to start my own career, I really wanted to do something that's different, at least thematically different from my graduate advisor and my PhD advisor. So I actually at the time, there's no internet. So I actually buried myself in the library for weeks just to read up everything. Yeah. And guess what I found? I found TF signaling. At the time, all these intracellular signaling proteins were just being discovered, cloned, by actually Vishwa Dixit was one of the early person who did a lot of work there. I said, okay, maybe that's what I would do. It's really interesting because this family of receptors can induce all kinds of different cellular responses. Sometimes it's death, sometimes it's kappa B activation, and how that is controlled really would depend on the intracellular signaling complexes. That's really how I started everything, and to some degree, that's also good luck, I suppose. That's also the beginning of, in my mind at least, beginning of innate immunity. So that's also when toll-like receptor were just cloned, and then it turned out TNF signaling complex overlaps with those that's used by toll-like receptors and interleukin-1 receptors. So that's my entry into the field. So I just became fascinated with all the biology that's there. So I feel like I'm still learning the biology. It's so complex. So I was at Weill Cornell for 15 years. And then, in fact, if I moved to Boston when my kids basically, when the kids were grown, then I was movable. So that was almost 10 years ago. So eight and a half years ago. So I started out doing structure, but I think as time goes, as I learn more about the field, I became really interested in the biology itself as well. We're looking for cell biologists who's interested in the field. We don't have too many in the lab. We only have a few, and I always feel that we have so many more questions that we want to address. For example, one of the things that we discovered was that these pathways, innate immune pathways, almost all of them form these higher order structures. They oligomerize, they interact with each other, they form these gigantic things in the cell. And that's actually when we started also doing cellular imaging. So we were sort of migrating from just high resolution structural approaches also to cellular imaging approaches. Now, of course, a lot of cryo-EM and also moving into cryotomography as well. So what else can I say? Oh yeah, so we also become really interested in therapeutics. I think these are all related. By the time you get into the biology and you feel that there would be biochemical skills that can help to develop drugs to counter all these devastating diseases. I started a company a few years ago, and it's doing pretty well. Trying to help to identify inhibitors or other types of modulators. So yeah, I think I should shut up now. Yeah, I think it's already too long.
Oh, well, actually, with the next question, I'm actually going to start with you. So you'll get to keep talking.
Okay, all right.
But I do want to talk, everyone seemed to have a pretty straightforward path and kind of stayed in academia. So I'm wondering if you can talk a little bit about why you chose to stay in academia, what sort of considerations you would take into account for those debating academia versus industry career paths, and how, since you recently started a company, maybe you can talk about some of the differences between academia and industry.
Yeah, actually, I think maybe about it's a decent fraction of postdocs from my lab goes to industry as well. So how can I start? So when I started the academic job, at that time, I just want to be a professor. There wasn't, I didn't really consider, even though I actually had an offer from one of the companies, I didn't go. I think it just, it's partly your interest. Now I know a little bit more, I think, about how industry works. At least just gleaning from what the company that I started are doing. There's really a huge amount of teamwork. I think you really need to enjoy that, working with others. And it can be extremely exciting because you just have one goal, and then you all go about it, to contribute your skills to that goal. It's different. It's different, but I think it can be equally interesting. If you have a lot of ideas of what project you want to do, you should go for academia, because then you have the flexibility to do that. But if you think you're really more of a skill person, you can do a lot of things, but you don't care exactly what questions you want to address. Industry is a great place to utilize your skills and make a difference. Yeah, I really don't see one above the other. I think they're equal. Just what you would enjoy better.
Great, thank you. Andreas, let's go back over to you. Can you talk a little bit about your considerations with academia versus industry, and what made you stay on the academia path?
Yeah, Jill, of course. In my personal life, the decision was not so much to go into a large company in the very first place. That came only in the very last years now. But as a clinician scientist, you of course have the possibility to go to a private practice, and just as a private practice nephrologist in Germany, you would earn just much more money. That's not the same everywhere in the world, but then the reason why I still didn't do it is just that I'm fascinated by these questions, and in my current view of how I want to lead my job, it's practically impossible outside a university setting. I would really need this interconnection with the clinical routine and the questions and the possibilities. As Hao said, you maintain your flexibility. That's, I think, the big plus for staying in academia. So if you have of course, it's tough, right? You need to be lucky to find something. You need to publish, and you need to somewhat finance your own lab. And if you're lucky in that, then this is it's a wonderful situation. But it can be very frustrating if all your grants get rejected. And I had earlier parts of my career where this was clearly the case. And where we didn't get a grant funded within 3 years, and we didn't get a paper out in 2 years, and really hardly anything worked. But then, if you stick to these ideas that you have, in our case, it's, really interfering therapeutically with regulated necrosis, which is clinically very important. And if you really want to bring that idea forward, then there will be a chance to somehow do it, I think. I mean, you have to believe in the idea. It's the general fundamental question that you have to believe in. And, then in the recent years, when the offers came in from larger companies, where you would have a much bigger footprint, of course, if you would lead a larger group, you would probably not have your own lab anymore. And as a clinician scientist, you would lose the direct contact to the patients. And that is, why I didn't do it so far. But of course, thinking about salaries in the companies, that's even better than being a fractional nephrologist. And I'm still on the very low side of that aspect, but I still enjoy it, right? And and I don't have any plans to change that either.
Great, thank you for sharing that. All right, Felix, can you talk a little bit about your path in academia?
Yeah, for me, it really was never a question. Once I realized in academia you really can do whatever you want, and somebody pays you for that, I was absolutely decided. There was no consideration after that anymore. But I can see that there is pros and cons on both sides. So on the academia, as I said, you can do whatever you want to do, but you really have to do it all by yourself, right? I mean, you have to be the driving force that has the ideas, that gets the money, that gets a team together, that addresses the problem. If you don't do it, nobody else will do it. On the biotech side or the industry side, I think it's probably a little bit more filling the space that you are given. You join a company, and that company has a certain goal. You join a team, and the team has a certain goal, and you do as best as you can within that project. And it can be very fulfilling, I think, working with other people, having shared goals, having a good working climate. That is probably very nice. And I could see myself in such an environment. That probably would work. The one thing I would be really, really struggling with is if a project is taken away. If somebody, a step above you or 2 steps above you, all of a sudden says, no, this project is going to be culled. And maybe not even a scientist, maybe somebody who has monetary power over the company, the finances basically decide that the project gets killed. I wouldn't like that. That's why I'm in academia.
Great, thank you for sharing that. The next thing we're going to talk about is some of the challenges you faced in your career and how you overcame them. Kind of talk about what worked, what didn't work. I know I've heard a little bit of some challenges from each of you, so you can kind of expand upon some of those, or maybe talk about something else. So Hao, would you like to go ahead and start?
Yeah. Challenges, yes, definitely. Let's see, what would be? I think one thing would be balancing between family life. Would that be part of what you're asking? Family life and work. It's actually really hard. what would be my advice? Yeah, I mean, try to think. I mean, do what you can, I guess. Yeah, partition. I can tell you, I guess, what I did. So I had my first child when I was in a postdoc. I just had to partition I had to work 9 to 5, in fact. Yeah, or a little bit earlier than that. I just come in, be the most efficient, do my work, and go home. And then at home, if there's a gap in the routine with the kids, then you do things that you can do remotely. You don't have to be in the lab. I think that happened even after I become a professor, that become a PI. I realized that I had to be very, very flexible. That is, whenever you have a gap, you can put work into it. That's what I had. It would be like a convoluted maze. Work is this shape, and then family is the other shape. But they're not like right cut in the middle. They're just completely meshed. And then you just do your best with the kids, and then whenever you have time, you work. And of course when you're off, you could think about your work. I don't know if that's a good model, but that's what I did.
Certainly family work balance is a challenge. I think
it's a challenge. Yeah, it's definitely a challenge. Yeah, right. You probably had to learn better. I mean, I don't think I did very well in that. That is, learn to say no to a lot of things, and really focus on the most important thing in your life at that moment. And that, of course, had to change constantly. You had to reprioritize constantly and make sure you don't get frustrated. Yeah, and enjoy every moment, whichever way. I think, like, bring your computer to the field if you watch soccer playing or something. Basically, you just have to be flexible. I would say, yeah.
Mm-hmm.
I can talk about other things, but that's all my sense for.
No, we actually often get questions about the work-family balance, so that's very good to address.
It's actually people are more aware of it. I would say when I was in it, I wasn't aware of it. I don't think I had a life, if you will, other than work and family. But I was actually okay with it. I didn't, my hobbies were all gone at that time. But I think I didn't mind, in fact. Yeah, just to have these two components so that it's a little simpler.
Great. Thank you for sharing that. Felix, do you want to talk about some of the challenges you faced and how you overcame them? What worked, what didn't work?
Yeah. So one thing that I find difficult in science actually is the ups and downs. We all have ups and downs, right? And you have your good days and your bad days, and you have and I think in science this can be amplified by things that you can barely control. A very recent example, last week we were, I was in a fabulous mood. We had published a very nice paper, I think a very nice paper. I tell you about it in 2 days' time and you make your own judgment. And I was in a very good mood. I was on a real high. And on Sunday morning, I woke up and started reading it around a little bit, and I saw that Neil Alto has published a paper in Cell that has scooped us absolutely 100%, and the whole story is gone. There really is nothing left there. And that's a real down. And one somehow has to live with these things and come up with strategies how to overcome them. And over the years, I've seen with some of my friends in science that had great trouble with this. I had a friend who had some bipolar problems, and everything got amplified by the scientific environment.
Mm-hmm.
But I have no real solution to this other than don't rely only on science. Have friends, have a family, have a hobby. Don't give up all the hobbies like Hao did.
All right, that's some great advice. Thank you, Andreas. Do you want to talk a little bit more about? I know you mentioned some challenges when you were talking about your path in academia, so I'm not sure if you want to expand on some of those or maybe talk about something else?
Yeah, I will probably answer this. I mean, there's a lot of things we can talk about, but maybe the two most important ones, just to not give the impression that everything's perfect. I'm divorced. That was clearly a consequence of working too much. This led to now driving every other weekend to see my kids for about 1,000 kilometers. I've been doing that for 8 years now, and my kids still love me, and I love them, and so everything's fine in that regard. The other thing is the hobbies. I wish I followed Felix's advice. I wanted to be a concert guitarist before I actually became a banker, before I started studying in med school. And so I was interested in a couple of things. I studied geology, studied sound engineering, and all of that is gone, right? There's the only two things in life that matter now is just the work in the clinic and the work in the lab. And I don't recommend this to anyone. It's not necessarily leading to satisfaction or to a wonderful life. But, out of this work, there's also a lot of positive aspects that come out of that. If you're good at frustration tolerance, this is the term that I always use. If you're okay with a paper being rejected and okay with a grant being rejected, and you just think, okay, what can I do? You have a beer and then you write the next application. That's pretty much all you can do. And I learned that from fantastic mentors, other scientists in our field like Peter van den Abele or Doug Green, who've been very helpful in these situations. And it's clearly not been easy. So yeah, and I wouldn't say, being asked to participate here in this session gives you the feeling that somehow people might think that things are perfectly fine now. They're not, right? So this is all just what comes to somewhat is a price of doing research in the way that you want to do it. That can draw you back a lot, I must say. So I'm not always happy with the job. I would not do anything else, but I'm not always happy with it. That's maybe a bit more depressing than compared to what the other two have said. But this is just what it feels like. It's still great to go to the lab and talk with the group.
Well, thank you all for your very honest answers.
Actually, there's this, maybe I can second that, delayed gratification. is a good skill to be in academia.
Yeah.
And maybe in life in general, yeah.
And there's this one thing that makes it so special. It doesn't happen very often, but every once in a while, one knows somebody that nobody else knows in the whole wide world, right? Except the person that will scoop you on a Sunday morning, right? But just discovering these things, that's really, really very nice.